hope you all had a great weekend. I miss all of you. What we're going to do today is we're going to wrap up this entire unit. So this unit was pretty long. It was about DNA replication and gene expression, but majority of it was about DNA replication, protein synthesis, right? And then leading our way up into what we talked about last week, which was gene regulation. But it was a pretty tough unit to start out with, considering that it was to start virtual learning with, considering that it was so long. But that's why I want to spend time, like before we move into our next unit, just wrapping everything up and making sure that you truly have an understanding on everything. But hopefully that, that little lesson on telomeres and you reading that article about Dolly and the cloning piqued your interest a little bit more about this topic. And then also our little discussion that we had about epigenetics and relating to, gene expression and gene regulation. I thought that was pretty cool and hopefully also made you a little bit more interested in the topic. The next unit that we're moving into tomorrow is genetics. And I'm so excited. I'm really sad that we aren't doing this unit together and we're doing it virtually, but hopefully still make it fun and interesting, and it's going to be a great time. What we're going to do is we're going to review the main points of what we talked about. I'm not going to get into like the nitty-gritty details of this eukaryotic gene regulation. I asked you to read these slides, so if you didn't, please do. The central dogma of biology. What is it? DNA is copied, right? What's that called? Transcribed into RNA, and RNA is translated into a protein. So remember we had that discussion about the different languages, right? DNA is a nucleic acid, which is made up of nucleotides, and then DNA is a nucleic acid also made of nucleotides, but a protein is made of amino acids. So that's why this translation part has to take place, right? So before we get into that, right, we also said a part of the central dogma is the fact that DNA also replicates. We have double helix DNA, and the first thing that happens, right, we have this enzyme here, helicase. It's going to unzip the DNA, and it's going to create that replication fork. So it'd all be review for you, right? That's horrible. But this replication fork. As helicase is unzipping the DNA, we have gyrase here, which relieves the buildup of torque as the DNA is unwinding, right? So we have this first enzyme that's going to lay down the RNA nucleotides. But first we have to distinguish the 5' end from the 3' end of DNA. We're gonna make this 3' to 5', and we'll make this bottom strand 5' to 3'. So in what direction does DNA polymerase 3 replicate? DNA polymerase 3 needs to replicate from 5' to 3'. Right? It needs to create the daughter strand from 5' to 3'. And since DNA runs anti-parallel, right, it's going to start at the parent strand's 3' end to build from 5' to 3', right? So down here would start from this 3' end to build from its daughter strand from 5' to 3'. So before it can do this, we need RNA polymerase. Specifically, the specific RNA polymerase is primase. Primase lays down the RNA primers, telling DNA where to start replicating. DNA polymerase where to start replicating. It's like a little start flag. Okay, so here, right, primase lays down a couple of RNA nucleotides, telling DNA polymerase 3 where to start. So DNA polymerase 3, make it blue, comes in and builds from the RNA nucleotides that were already laid there, right? This is a daughter strand, so it's not actually connected. It's connected through hydrogen bonds, and it's building. The primase is essentially laying down those nucleotides in thin air because DNA polymerase 3 can't lay down the nucleotides by themselves. It needs something to build off of. Now, this top strand would be called the what strand? It would be called the leading strand because DNA polymerase is building the daughter strand in the same direction as the replication fork versus this bottom strand, which would be called the lagging strand because DNA polymerase 3 is building it in the opposite direction of the replication fork. So, right, once the DNA polymerase reaches the end of this strand, right, well, it's going to have to build the rest of the strand. So RNA polymerase, primase, is going to need to make more primers as the DNA unzips, right? So you're going to have these sections of DNA. DNA is being built now in sections, and those fragments are called the Okazaki fragments. So that's the difference between the leading strand and the lagging strand. Now the next enzyme that comes into play, we'll make it green, is called DNA polymerase 1. DNA polymerase 1 is replacing the RNA primers with DNA. So it's going to have to replace these RNA nucleotides all the way down here on both strands with DNA, because RNA has uracil, right? DNA has thymine, RNA has ribose, DNA has deoxyribose, so they need to be replaced. Now, once they are replaced, right, well, there's going to be a little breakage in the backbone of DNA in between the new DNA nucleotides and that were replaced that replace the primers, and also in between these Okazaki fragments. And that's the last step, is ligase, which is going to glue those fragments together and does that by filling in the gaps with phosphodiester bonds, the bonds of that backbone. So DNA replicates in a semi-conservative way, which some of you did draw incorrectly on your diagrams. So our parent strand was purple. Our two parent strands were both purple, and our daughter strands are both, we'll use that blue color. Semi-conservative means we started with this DNA parent strand, two purple, and it's semi-conservative, so half of the parent DNA stays, and then the other half would be that new daughter strand that was replicated. So you end up with two DNA, one of each DNA has the parent strand, and the other one has one of the newly replicated daughter strands. Now, one thing that we just added this week was talk about telomeres. As this lagging strand, right, keeps going, it's going to reach a point near the end of the strand, right, once it gets to the end of the strand of the daughter strand. Up here, this 5' end of the daughter strand, of the lagging daughter strand. Oops. So as this keeps replicating, right, eventually it will get to the end, so I'll draw it down here, right? So you're going to have the daughter strand, and you're going to have the parent strand, which was purple, and the replication fork was going this way. So once you reach the end of this daughter strand, the 5' end of the daughter strand, there was a primer, right, telling DNA polymerase where to go, and the DNA polymerase built in that direction. So after this primer, this RNA primer has been removed, the DNA polymerase has no existing nucleotides to add onto to replace that primer, since it's at the end of the entire strand. There's nothing over here to help DNA polymerase build the primers to replace the primers with DNA, because it has to go in the 5' to 3' direction. There's nothing on this side.
There is no 3' hydroxyl group that's going to prime the DNA synthesis.
So that is going to pose an issue, because that means that at the end of the one side of the DNA, it would get shorter every time, because that lagging strand is built in segments, and it wouldn't be able to get rid of, to replace the RNA primers with DNA, because there would be nothing to hold on to, so it would just get rid of the primers. So it's going to be shorter. So that's why we have this enzyme called telomerase. So it lays down telomeres, and telomeres are repeating non-coding segments of DNA that are going to go at the 3' end of the parent strand, and it's going to add on a whole bunch of non-coding DNA. That's going to ensure that the overhang of the parent strand is not a coding sequence, and it doesn't matter if it gets replicated or not on the other side. So this will allow the daughter strand to be able to copy the information that it needs to, and then the primers that code for this nonsense are going to not be able to be replaced, and that's okay. We talked about pair mechanisms, right? So DNA, in order to prevent mutation, and during this replicating process, there can be some errors. Both DNA polymerases, DNA polymerase 1 and DNA polymerase 3. So we have these two different polymerases. We have DNA polymerase 1 and DNA polymerase 2. And they basically both take part in DNA repair. So DNA polymerase 1 is going to be the polymerase that replaces the RNA nucleotides. This is also the enzyme that repairs DNA after replication as well. DNA polymerase 3, that's the one that lays down the DNA nucleotides in the 5' to 3' direction. It proofreads while it's replicating. So like checking, making sure that it's laying down the right bases. I realized that I missed a base or I put the incorrect base, and it could proofread and then, correct its mistakes. It could also do some repair after DNA replication as well. So remember, we talked about the couple different types of repairs. Mismatch repair is what happens during DNA replication. That's its proofreading. If it realizes it, put an incorrect base, that it could use 3' to 5' exonuclease activity to just remove it. After replication, base excision can take place, or nucleotide excision, and that would be done by DNA polymerase 1. So, cutting out the incorrect base and replacing it with the correct one, cutting out an entire sequence of bases. And then also, nucleotide excision is when you have that thymine dimer that can be, cut out that damaged region and put in a new undamaged region. So that moves us into protein synthesis, which is part of our central dogma of biology. So protein synthesis, you have 2 strands of DNA. One strand is going to be called the coding strand, while the other strand is known as the template strand. Remember, there's a couple of names for them. So you can call it the template strand or the antisense strand, and then this is the coding strand or the non-template strand or the sense strand. So the first step of protein synthesis is transcription. So step number 1, this is where DNA is copied into mRNA, and this is happening in the nucleus. Now, the coding strand is what has the information has the code sequences, the genes that are used to make proteins. So we need to have the genes in our mRNA strand. How are we going to do that? By copying the template strand, because through complementary base pairing, the copy of the template strand is going to have the codes from the coding strand. So the template strand is copied in order to create the same codes as the coding strand. So transcription happens in 3 steps. Step 1, initiation. RNA polymerase is the enzyme in charge for protein synthesis. So it's going to attach to the promoter sequence of DNA on the template strand, of course, which is a sequence specific to starting off transcription. So it's like, the Tata box. Thymine, adenine, telling RNA polymerase where to attach to, the promoter sequence. This is initiation. The second step is called elongation. This is where RNA polymerase is going to unzip the DNA, separate the strands, and lay down RNA nucleotides via complementary base pairing. So it's going to, the initiator sequence is where transcription actually begins, right after the promoter sequence, and the G coding sequence. That's the first sequence of DNA. That will be a template for the initiator codon. And elongation is when RNA polymerase is copying those sequences from the template strand. Okay, then finally, termination, which is the third step of transcription, which kind of tells you what it does. It comes off. The RNA strand separates from DNA. DNA recoils into the helix. So now the mRNA needs to get prepared to leave the nucleus. So it gets prepared through that process called RNA processing. So remember, you need to add the cap. Splicing needs to occur, which is removing the junk DNA, the introns, so that way the exons are there and they're going to get stitched together. The exons are what's becoming expressed. And then polyadenylation occurs, which is the addition of the poly A tail. Right? So what's the purpose of capping, adding that cap and adding that tail? The purpose is to protect the exons, because those are the actual genes that are going to be expressed. We want them to be protected. So that's why we're going to add that cap and we're going to add that tail, because this mRNA is going to be leaving the nucleus And we said that there's enzymes there, RNA ACEs, that are there to destroy mRNA. We don't want our exons to get destroyed, so we have some extra protection.
That leads us into our final step of protein synthesis, which is translation, and that takes place in the ribosome.
With some transfer RNA, some tRNA, and some rRNA that are in the ribosome. So you have your ribosome here, which kind of looks like a fish. The mRNA gets fed into the ribosome 5' end first, so moving in that direction. And there's these different sites inside the ribosome: the A site, P site, and the E site. So first step, initiation at the A site. The mRNA codon is read, and the tRNA that has the complementary anticodon is going to bond to it. So the tRNA has the complementary anticodon here attached to the tRNA on the other side. To know the amino acid that corresponds with the codon, the 3 nucleotides on the mRNA strand. After the A site, the mRNA is going to move, again, 5' to 3', so it's moving in this direction. And this tRNA follows the codon and moves to the P site. So the tRNA moves from the A site to the P site for this elongation step. This is where the amino acid chain is going to get built. And then the tRNA would, leave its amino acid behind to the next tRNA that moves to the P site. That tRNA is going to move to the E site where termination takes place and the tRNA leaves. The amino acid chain being built in the P site. So a whole bunch of amino acid chains built together. That is what creates your protein. So there are a couple DNA mutations that can occur that can affect the protein. So we talked about point mutations and frame shift mutations. So some point mutations or substitution mutations, substituting a base for another base can lead to a silence mutation, which means that no change occurs. A nonsense mutation, which it's going to call for a premature stop codon, or a missense mutation, which an example of a missense mutation would be sickle cell anemia, and that would lead to a base pair change with a change in the amino acid. Frame shift mutations, that could be an insertion of one or more bases or deletion of one or more bases, which is then going to change the codons, how they're being read, which is going to change the amino acid sequence in the resulting protein. And that leaves us off with gene regulation. So we first started talking about gene regulation with prokaryotes. So just to remind you, the purpose of gene regulation in both prokaryotes and eukaryotes, right, is being able to turn genes on and off. If we want a gene to be expressed, well, then it's going to turn on. If we don't want it to be expressed, then it's going to turn off. So for example, right, the Amoeba Sisters video talked about how all the cells in our body, all of our body cells, our somatic cells, most of them, with some exceptions, like red blood cells don't have a nucleus, they don't have DNA, but most of our body cells have a nucleus. If they have a nucleus, they have all of our DNA, whether it's coding for a protein that belongs in our stomach or belongs in our eyes, they all have all the DNA. So that's why we have this gene regulation process to turn genes on if we want them to be expressed in that cell, or our body will turn them off if they don't need to be expressed or they shouldn't be expressed. Now, we first started talking about prokaryotes. Prokaryotes have operons. So the expression of genes in prokaryotes is regulated by an operon. So remember, that includes the promoter region of DNA where the RNA polymerase is actually going to bind to, the operator, which is the region of DNA that regulates certain genes from being expressed, the repressor, which is the protein that binds to the operator, and this allows the genes to be regulated. The example that we went over was the lac operon. So this is in prokaryotes, right? Bacteria is an example of a prokaryote. They do not have a nucleus. So if you look at this strand of DNA, and this obviously is something that takes place during transcription. This prokaryote does not want this gene to be, whether it wants the gene to be transcribed or not, they have this regulation system. So in the DNA, the promoter region, that's where the RNA polymerase is going to bind to. And right after the promoter region is this operator.
This is where the lac repressor would bond to.
And that's going to inhibit the RNA polymerase from transcribing this DNA sequence. So if the operator is binded to, if the repressor is binded to the operator, then RNA polymerase will not transcribe the gene. But in this specific example for the lac operon, the lactose, if the lactose binds to the repressor, which is a lac repressor because it's specific for lactose, then the repressor will move away, telling the RNA polymerase that it can transcribe this gene to make more lactase. So lactase is an enzyme that breaks down lactose. So if this is showing that there's a presence of lactose because it's binding to the receptor and moving it away, it's activating this transcription to code for more lactase to break down that lactose. So then once the lactose is broken down, right, then the repressor binds to the operator again because it's saving energy. It doesn't need to be producing lactase unless there's lactose there. If there's no lactose, then there's no reason to make any lactase. Another example was the trip operon. Now, eukaryotic regulation. So one that your video talked about was chromatin accessibility. So the structure of chromatin can be regulated. So remember, chromatin, unorganized DNA and proteins. The more open or relaxed a chromatin, it makes a gene more available for transcription. So it allows the gene to be transcribed so it can be expressed. The addition of these specific chemical groups, the acetylation, acetyl group, a methyl group, methylation, or phosphorylation, the addition of these chemical groups to histones,
which are the proteins that DNA wraps around,
lowers their affinity towards the DNA, is going to allow the DNA to unravel into loose chromatin so that way it could be transcribed.
When DNA is wrapped tightly around the histone,
not allowing them to unravel into the loose chromatin, well, then it's not going to be transcribed. So that's an example of one that happens during transcription. But make sure, like I said, you review the other ones that we talked about. I don't want to spend too much time talking about it right now because I wanted to just leave off with that little piece about epigenetics. Basically, nature and, how and our life experiences and what we go through throughout our life can change how our genes are expressed. It can change and add methyl groups to our DNA, like that little animation showed. So it was interesting, that video where you have twins who are genetically identical, but through epigenetics and personal life experiences or life choices, the environment can change how a person's genes are expressed and then can actually make them, their DNA is still the same, but the portions of DNA that are expressed are different. Do that assignment about epigenetics because it's something that we don't spend a lot of time on, but it's pretty interesting how gene expression, can be related to, something like that. And now, especially because we're moving into genetics, I just thought that was a great way to kind of segue us into genetics. So I know this was kind of long and maybe seemed rushed, but hopefully it allowed you to reflect on the main points of what we learned from this unit because it was extremely long, and kind of bring them out to you. And then hopefully it allows you to reflect on it, and then you'll be able to join the Zoom call during extra help hours and ask any questions or review. If you don't have any specific questions but you want me to review something, I'll be available. Or if you just want to, like I said, pop in, say hello, you can do that as well.